Hello everyone, this is the fourth and last video of the series, Are Humans Still Evolving? And in this video, we'll be looking into the HLA-B4601 allele and its role in human evolution in response to infectious diseases, specifically leprosy and HIV. Now, the LHA-B4601 allele arose approximately 50,000 years ago in the Upper Paleolithic era during a time when Southeast Asians first encountered leprosy introduced from Africa. Now, I think that data is worth taking note of. Now, leprosy, also known as Hansen's disease, more colloquially, is a long-term infection by the bacterium Mycobacterium leprae, which leads to the damage of the nerves and the respiratory tract, the skin, and the eyes. Now, symptoms of the disease include the discoloration of skin, thick or stiff or dry skin, and the curling of toes or fingers, as shown in the image right here. Right, that's leprosy. And moving on, the HLA-B4601 allele is a product of the genetic recombination between two parent alleles. First, the LHA-B1501 allele and the LHA-C0102 allele. Now, this recombination event, also termed as the intergenic mini-conversion, produced a novel allele that was particularly effective at presenting peptides from the Mycobacterium leprae to the immune system. Now, more specifically, the LHA-B4601 allele contains the C1 epitope, which is specifically recognized by the KIR2DL3 receptors on these NK cells, also known as natural killer cells. Now, these NK cells are super important because they play a crucial role in the immune response by targeting and destroying infected cells. Now, the interaction between the HLA-B4601 and the KIR2DL3 helps boost the immune response against the Mycobacterium leprae, thereby potentially increasing resistance to the disease. Now, the ability of the HLA-B4601 to effectively present pathogen-derived peptides makes it a critical component of the immune surveillance apparatus. Now, by presenting the leprosy-derived peptides more effectively than its parent alleles, the HLA-B4601 provided individuals with a survival advantage in regions plagued by leprosy, which led to an increase in its frequency in the population. Now, we can really verify how this is true by examining this map here. Now, the map here shows that in Southeast Asia, the concentration of the allele correlates with the concentration of the leprosy patient cases. So we really do kind of recognize that sort of trend here. However, the specialization of the HLA-B4601 comes with trade-offs. While it adapted to combat leprosy, it does not necessarily confer the same level of protection against any other pathogens. For instance, the LHA-B4601 allele can increase the risk of HIV infection by affecting the immune system's natural killer, the NK cell immune response. Now, unlike the other HLA alleles that enhance the HIV-specific T-cell responses, the HLA-B4601 contains an HLA-C epitope, which binds to inhibitory receptors of the NK cells. Now, this interaction inhibits the NK cell's usual function in usually controlling the HIV infections. Now, this case study was a clear illustration of how, despite a susceptibility to one disease, in this case the HIV infection, an allele may still be prevalent due to another disease that is even more severe in the region.